In this lab, we'll be discussing the anatomy of the neuron. So if you recall, when we were doing tissues, the nervous system was one of the four primary tissues. We had briefly looked at the neuron, and we saw that it had three parts. The first part was the dendrite. The second part we looked at was the cell body. And the third part was the axon. Today, we're going to look at them in a little bit more detail. This is a picture from our book that shows a neuron, and just to kind of show you this whole circular area here, kind of where my cursor is, would be the cell body or the soma. Then these fibers here would be the dendrites. They're attached on this neuron. They happen to be attached to the soma. Then over here, this is the axon, right? The longest part going away from the cell body. So just as a reminder, for lab, you need to be able to identify where these structures are on a model or a photograph. For lecture, you need to know the function. So let's take a look at the dendrites. The dendrites, as far as function, think of as the receiving portion of the neuron. So when information is entering the neuron, we see that it enters the dendrites. So again, all of these fibers attached to the cell body on this neuron, because it's not always the case. We're going to see dendrites can be elsewhere, but on this neuron, it happens to be on the cell body, and they're the receiving portion. The cell body contains all the organelles that keep the neuron functioning. I put here mitochondria, the Golgi, the lysosomes, all the normal things, nucleus, the nucleolus, everything that we would expect to see. They're going to be inside the cell body, and you can see them all kind of over here is the nucleus and the nucleolus. But something unique to the neuron, the ribosomes, when they produce proteins, they cluster up, they clump up. We can see here's one, a little clumping here. All of these little clumps are ribosomes on the ER, and they're producing proteins when the neuron is growing or the neuron is repairing itself if it was damaged, that type of thing. So we call these ribosomes the Nissl bodies. That's all. You can see the spelling here, Nissl, right? Not Nissel. The correct spelling is Nissl. Right? So those are just clumps of ribosomes on the ER when the cell is producing proteins. The last structure I want you to be able to identify on our models, if you take a look here, these little fibers running through the cell body, you can see them all over the place. These little fibers are actually part of the cytoskeleton. Similar to what we saw when we did the anatomy of the cell, there's a whole skeletal system that kind of supports the cell. We're going to call these filaments here, or these fibers, neurofilaments. They clump up or cluster up to form neurofibrils. So on an exam, I would be okay if you said neurofilaments. You could call them neurofibrils, or even easier, you could just say the cytoskeleton. But their function, again, is going to be to support our cell. So here's the neuron model. This is what I call the white neuron. We're going to be using two models in lab. This one's the white one and the other one's the yellow one. So you can see here, as far as the soma, all of the soma would be here. Within the soma, we have the nucleus. The little blue dot inside is the nucleolus. We can see some nuclear pores. They're not showing any mitochondria or anything like that, but they do show these clusters of the proteins. All of these clusters here are the Nissl bodies, right? So on this model, the Nissl bodies are all of these blue dots. Then here, these black lines, this happens to be running in a dendrite. These black lines would extend throughout the cell. These are the neurofilaments. Again, when they bundle up, they form the neurofibrils, or you could just call it the cytoskeleton. The dendrites on this model, guys, are these gray structures here. So all of these gray projections would be the dendrites. Here we see one over here. Ignore these orange things. We're going to get back to that in a couple minutes. On the yellow neuron, again, the yellow projections would be the dendrites. This is what we had looked at when we were doing this, the tissues. Here we can see the nucleolus and the nucleus. Let me blow it up a little bit. Now, this model here, the Nissl bodies are there, but they're white, so they're a little bit hard to see. So somebody seemed to have did a little bit of an outline here. Somebody outlined this one, but you can see if you look up closely, when you look at the model, you can see these little white areas. Those are the Nissl bodies on this model. So it's not quite as easy to see as it is on the white model. What I do like about this model, though, the cytoskeleton, right, the neurofilaments and the neurofibrils, you can see them much easier. All these brown lines here are the cytoskeleton. Right, so we got to know both of them. I use both of the models. One shows one thing better than the other. Okay, so those are the structures of the cell body. The axon. So the first landmark where the axon meets the cell body, this kind of cone-shaped area right here is the axon hillock. Then just distal to it right here, this is known as the initial segment. This is where the first action potential or nerve impulse is generated on the axon, and then that action potential is conducted down this way. You can even see here in your book, they drew that red arrow going this way, showing you which way the action potential is going to travel. So the first part is right here. That's known as the initial segment. The cell membrane of the axon, if you kind of follow my cursor here, all of that cell membrane we call the axolemma. The cytoplasm that's running down the axon, right inside the axolemma, all of this blue here is going to be called the axoplasm. All right, so those are the structures of this part of the axon. Okay, so we have the axon hillock is the joining segment. The initial segment is right after the axon hillock. Very simply, the cytoplasm of the axon is the axoplasm, and the cell membrane of the axon is the axolemma. So on this model here, we could see this joining place here, this kind of cone-shaped area is the axon hillock. Then just distal to it, kind of a general region right here, this would be initial segment. And then we could see here's the axon. Now on this side is the rest of the axon. Over here, they cut away the axon so we could look inside. So all of this would be the axoplasm. And you can see there's some mitochondria in there. The blue dots will be the Nissl bodies. And then all of this little edge right here is showing the axolemma, right? So all of this gray is the axolemma. I tried to put my arrow on the edge so you can see the edge of it. Okay, so let's finish up the axon. So an axon, when it ends, doesn't end as a blunt end. It actually ends as as branching. Let's find that picture again. So if you take a look over here, if we follow this axon, it ends as all of this branching over here. These branching, you're going to call the telodendria. By having the telodendria, the axon can touch more than one structure. It's able to innervate more than one structure. So it's kind of an important function. Then at the end of the telodendria, you're going to see a little bit of a swelling, right? These little swellings right here. Our book labels them as the axon terminals. There's a lot of different terms for them. I put some other ones here. Sometimes we call them the synaptic bulb. Some people call them the end bulb. Some people say the synaptic end bulb. There's all different variations. What's important about that, inside the synaptic end bulb or the axon terminal, are these chemicals called neurotransmitters. And the neurotransmitter is necessary for conducting our nerve impulse, the action potential, to another structure. We'll see that in lecture when we talk about synapses. Some of the neurotransmitters you may know are acetylcholine, dopamine, serotonin, epinephrine. There's a bunch of them. Those are the ones you may have heard of. Okay, so the branching we're going to call the telodendria. The swellings here are going to be the axon terminal. Now, on our model, let's find it. If we were to follow this axon down over here on our model, we would see it ends as a blunt end. They didn't show the telodendria. So this is the way this model company did it. If you look up over here, here's a telodendria, here's an axon terminal, but it's not part of this neuron here. It's actually from another neuron coming to synapse with our neuron. So you can see I labeled this the telodendria of a different neuron coming to synapse with our neuron. Here's the axon terminal again of a different neuron, of other neurons coming to synapse. So they're actually showing it from a different perspective. If I go back to our picture here, it would be like this. Let's just say here's our neuron, the purple one. Then if we would have just kind of cut this off right here and just show the axon terminal and the telodendria of this blue neuron synapsing with the purple neuron, right? This is kind of what we would see on our model. We would only see that part here. And so they're showing it from a different perspective. All right, so you got to make sure you're understanding that this is the telodendria, again, not of this white neuron, it's a telodendria of a different neuron and the axon terminal. So it's just a different way to do it. The last part of the axon that we want to speak about is something called the myelin sheath. This is actually a cell that wraps around the axon. If you look at this axon here, this one is bare. It does not have a myelin sheath. We would say this one is unmyelinated. But take a look at this picture. If we were to follow the neuron here, here's the cell body, here's the axon hillock. If you follow the axon, the blue tube running down the middle here, all of this blue tube running down the middle, that would be the axon. But if you notice, this axon has these kind of hot dog buns on them. That is the myelin sheath, and that's actually a cell called the Schwann cell. That Schwann cell, if you look at your picture, it'll be a little bit clearly in your book, wraps around the axon. You can see there's little wrappings right here. Over and over again, up to 100 times, the Schwann cell will wrap around the axon and kind of insulates it to speed up the nerve transmission. So each one of these cells you can call the myelin sheath, or you could call the Schwann cell. Also notice this little bump out here. Each one of the Schwann cells has a little bump. When the Schwann cell wraps around the axon, the nucleus stays out on the periphery. So this little bump actually represents the nucleus of this living cell, the Schwann cell. All right, so you can't just say nucleus on an exam. You'd have to say the nucleus of the Schwann cell, because we already have a nucleus of the neuron up over here. All right, so you got to realize that there's two different nuclei. Also notice the Schwann cells don't touch one another. There's actually a gap. That becomes important. We'll talk about that in lecture because it speeds up the nerve impulse. So for today, your book, if you notice, they just call it a node, but we're going to call it the node of Ranvier. It's kind of an older term, but most people seem to remember it. It's the node of Ranvier, and it's just the gap between the Schwann cells, right? They should not touch. It's a microscopic gap. Finally, the cell membrane of the Schwann cell, right? So all of the cell membrane, because these are just regular cells, this we're going to call the neurolemma. You'll study this more in pathology. This is something that's important when a neuron is severed and it regrows. The neurons in our peripheral nervous system have the ability to regrow and repair themselves. That neurolemma becomes important. So I just put here it's the cell membrane of the Schwann cell, or here I put the outer layer of the Schwann cell. So let's take a look at it on our model. So all of this cell here on the axon is the Schwann cell. I labeled it such, and you can also call it the myelin sheath. If you look at it closely, you'll see these lines going up and down. This actually represents the wrapping, how the cell wrapped around the axon. This here would be the nucleus of the Schwann cell. Right, here's the other Schwann cell on this model, right, the nucleus of that Schwann cell. And then right here, guys, the gap. That little squiggly line represents a gap between the two Schwann cells. That's known as the node of Ranvier. And then here, the cell membrane is known as the neurolemma. Okay. We got that done. I labeled it all on the yellow neuron also. So if you recall, a couple seconds ago, we said that our model shows the telodendria and the axon terminals from other neurons synapsing with our model. So I want to show you the three different places that neurons can synapse with each other. The first synapse is known as an axodendritic synapse. So if we go back to our white model here, if the axon of one neuron, right, so again, we said this was the telodendria and the axon terminal of another neuron, synapses with, let's say right here, the dendrite of our neuron. This synapse here would be an axodendritic synapse up here, right? The axon of one neuron here synapsing with the dendrite of our neuron. This would be axodendritic. The second possibility, if an axon synapses with the cell body, that would be an axosomatic. So if you take a look over here, here's the axon of the other neuron synapsing with the cell body right here. That would be axosomatic. This looks like it would be another one too. This is kind of going to the side of the body. So this would be an axosomatic synapse. The third possibility is an axonic. So here, the axon of one neuron synapsing at the axon hillock of the other neuron. So this would be axon to axon. I did it also on the yellow neuron. You can see over here, this would be an axodendritic. Right? This looks like it would be an axodendritic. Here's an axosomatic. Notice on this model company, they only put the axon terminal down. They don't have the telodendria. All they're showing you is the axon terminal. Also notice this one here, there's no axonic synapse, right? All they have is the axosomatic and the axodendritic. Finally, the nervous system or the nerves have connective tissue coverings. In lecture, we're going to talk about the epineurium, the perineurium, a little bit more detail. But just to kind of go over it, if we took a nerve, let's say we took out the sciatic nerve, which we had looked at when we studied the muscles of the hip, the covering around the entire sciatic nerve would be called the epineurium. Then if we chop that sciatic nerve in half and looked into it, we would see that there's these bundles that are called fascicles. And within the fascicles are anywhere to 10 to 100 axon fibers. But surrounding the fascicle is the perineurium. Then surrounding each one of the axons, so the axon is covered, is going to be by the endoneurium. Right, here's a picture from your book. Like, let's say we took out a nerve, like the sciatic nerve. Right, so the covering around the entire sciatic nerve would be the epineurium. Then when we look in, these purple bundles right here, those are the fascicles. So that purple connective tissue would be the perineurium. Then the circles within, all of these circles are the individual axons, they're going to have the endoneurium. I like this picture. They pulled out one of the fascicles, and you can see the purple there is going to be the perineurium. Then they pull out a axon, and the axon is going to be surrounded by the endoneurium. Right, so that's going to be surrounded by the actual endoneurium. Right, so that's, you can see that's on the Schwann cell. So in the lab, all we have is the axon with the endoneurium. We don't have any. We don't have any models that show the peri or the epi. So notice here, this is a little piece of connective tissue. This is going to be the endoneurium. This would actually go up and cover the entire neuron. Right? So this is going to be the endoneurium. On the yellow model, we can see it a little bit better, right? Here's the endoneurium. Again, they cut it so that we could see all the structures here, but the endoneurium would cover all of the axon. And that's the end of our neuron anatomy lab. Our next recording will be of the spinal cord.